Hey y'all. In this metabolism project, we will be going over ATP as cellular currency and types of phosphorylation. So ATP is known as adenosine triphosphate. It's going to be the most broadly used chemical currency out of all the cells. It can be used for different things such as biosynthesis, mechanical work, including molecular transport and flagellar movement. The main components of ATP are going to be two phosphoanhydride bonds that are going to be connected to a ribose sugar with an ester bond. Then there's going to be the ribose sugar and an adenine base. So the three forms that this adenosine phosphate can take will be adenosine triphosphate, diphosphate, and monophosphate, and they're only going to differ depending on how many phosphates are connected to the ribose sugar. So how cells will use ATP is they will be breaking the phosphoanhydride bonds on ATP, and what that's going to do is transfer a phosphate group that will drive reaction. So you can transfer up to two phosphates, but adenosine monophosphate cannot transfer phosphates easily because it's a very hard bond to break, that ester bond. And in metabolism, cells will make enzymes to convert environmental elements into ATP. So basically what they're going to do is catabolic reactions are going to make ATP that the anabolic reactions will use. And this means that it's just like money, it's just like currency that we use. So ATP will be used by cells to produce cellular components. Similarly, like with us, it's like us using money to use and buy things to obtain different items. So ATP hydrolysis, so this is what the cells are going to do to use the ATP. It's going to give the product of adenosine diphosphate and inorganic phosphate. And these byproducts can be used to make more ATP. So the reason that this happens is that ATP will have a greater potential energy than its products produced after hydrolysis, which will lead to a negative delta G, and a negative delta G will drive the reaction forward, making it energetically favorable. And it can be used in an enzyme-coupled reaction, which we will go over a little bit later. So the enzyme-coupled reaction, for example, for the metabolism of glucose, with no enzyme-coupled reaction, ATP hydrolysis would just release all energy as heat, and the reaction would not occur, and you can see this through the positive delta G. But if the reaction would be coupled with the hydrolysis of ATP, the reaction will move forward, leading to a negative delta G. That's going to show that the reaction is energetically favorable, and a phosphate from the ATP will be given to glucose. This is just showing that coupling an enzymatic reaction with ATP hydrolysis could make it energetically favorable and more likely to produce more ATP in the future. So how we make ATP is going to be, first one we're going to talk about is substrate level phosphorylation. It is an enzyme coupled reaction, and it's going to make ATP by transferring a phosphate from a catabolic intermediate to produce ADP. The intermediates will include 1,3-biphosphoglycerate and phosphophenylpyruvate, or PEP. In glucose metabolism through substrate level phosphorylation, you're going to have 4 ATP made, 2 from the TCA cycle and 2 from glycolysis, and this is mainly going to occur in chemoorganotrophs in the cytoplasm. Now, oxidative phosphorylation will do things a little bit differently than substrate level phosphorylation. They're going to use the energy of a proton moving through an ATP synthase to make ATP. It's going to use a chemiosmosis model where electrons are going to be moved to the membrane and carried by NAD and FADH2, and this is going to give the energy to pump protons through that ATP synthase to make ATP. And this is going to occur in chemotrophs in the cell membrane. And for glucose metabolism, you're going to have 34 ATP made with oxidative phosphorylation. So this is just a picture found in the book on page 182. It kind of gives you a visual on the substrate and oxidative level phosphorylation, showing that oxidative phosphorylation is going to bring hydrogen outside of the cell, back into the cell wall at the membrane to make ATP, whereas in substrate level phosphorylation, you're going to have them all inside the cytoplasm. So this type of phosphorylation is going to be photophosphorylation. These will also use chemiosmosis, and it's going to be a lot of cells using photosynthesis where they will use light energy to make chemical energy. And very similarly to oxidative phosphorylation, they will use a proton motive force to fuel an APC synthase to synthesize ATP. And this is mainly going to occur in photostrophs.